can tell us something about brain activity in that region, talk a bit about neurochemicals and particularly MR spectroscopy and how we can combine these two to understand about the neurophysiology of the changes. Talk a bit about how we can combine them to understand connectivity changes within the brain. A little bit about the specificity of any brain activity changes. So rather than just looking at bold, can we go a step further and start to understand not only which brain region is active, but possibly which types of neurons are active and what that might tell us. And then finally go on to the how and talk a bit about the setup of transcranial electrical stimulation, so alternating current or DC in the scanner. And perfect timing. Hello, darling. I'm halfway through a lecture, sweetheart.
Are you?
Yes. Can you say hello to everybody and then go away?
Hello.
Thank you. Goodbye.
Hello.
Perfect. Welcome to my world. Sorry. So yes, we'll talk a little bit about how we can set these things up, what the major pitfalls are, how we've overcome them, and some suggestions for how you can start to get going if it's not something you already do. Okay, so brain activity. So one of the questions we had a little while ago, and again, I'm going to use this as an exemplar of the kind of questions you can ask with TDCS and brain stimulation and scanner. You may well not be interested in the actual experimental question. I hope you are. I hope it's interesting. But even if you're not, then hopefully it'll give you some ideas that you can start playing with to try and understand a bit more about brain activity. So the question we had then here was, we knew at the time, or thought we knew, that TDCS can improve behaviour in chronic stroke. So if you stimulate either the lesioned hemisphere or the unlesioned hemisphere with TDCS, you've got behavioural improvements. The question we had then was, how does that happen? Can we combine brain stimulation and neuroimaging to look at changes in brain activity? So what we did was take a group of chronic stroke patients and pop them out with a very old 3 Tesla scanner at FIMRIB. It should be in the Science Museum these days. This is what it looked like at the time. And we asked stroke patients just to move their wrists. So they did a wrist flexion extension in a block design, 30 seconds on, 30 seconds off. And they did that before and after either anodal stimulation to the lesioned hemisphere, cathodal stimulation to the unlesioned hemisphere, or sham. And the questions really were two specific questions. One was, what brain changes do we see associated with behavioural improvements? And also, can we use this not only to look at kind of group level changes, but can we use brain stimulation and MR to look specifically at brain activity changes underpinning behaviour and ask some causal questions? We'll get a bit closer to that anyway. So these are the behavioural results. So this was follow-up work, and you can see we used, so this is reaction times. So improvement in function is shown as a decrease in reaction time, so downwards is better. And in green is anodal stimulation to the ipsilesional hemisphere, and you can see that they got significantly faster reaction times. Cathodal to the contralesional hemisphere was something that was then and still is something that's suggested in the literature, in that you downregulate or decrease excitability in the contralesional hemisphere, and that increases excitability on the other side, and that leads to behavioural effects. This was the hypothesis. We didn't see any behavioural changes, but we in this group, and there are, this is not a stroke talk, so I'm not going to go into that in massive details. And we saw a slowing with sham, as we always do. We're looking at time and changes over time, and patients get very tired. So we repeated those that behavioural tests in the scanner and saw pretty much the same pattern of results. Again, an improvement with anodal stimulation, no change really with cathodal, and a worsening of behaviour with sham. And then what we wanted to look at was, can we look for regions of the brain that are active after stimulation compared with before, when people are moving their wrists, compared with sham stimulation? And what we saw was a pattern of activity in the motor areas of the brain, whereby we got more activity when patients moved and flexed their wrists after anodal stimulation compared with before stimulation, compared with sham control. So you can see that you get increases in these areas that are significantly increasing in red yellow, and you can see areas in the supplementary motor area, the premotor cortex, the motor cortex. The next question we had was, okay, fine, but how does this allow us to, how does this compare with cathedral stimulation to the other hemisphere? Even though we don't see any behavioural effects, does the pattern look similar? So this is the same contrast, but with cathedral stimulation to the other hemispheres. You can see they're stimulating here this time, and they're moving their stroke-affected hand. So we're looking for changes in activity in the stroke-affected hemisphere. And we do see them. So we've got these two different patterns of activity that lead to actually statistically indistinguishable maps. And that's all well and good. And you could do that actually really with any technique, right? So you could use any kind of intervention and say, okay, which areas of the brain are active when they do this task before our intervention, which areas of the brain are active afterwards, and look for changes. But the really interesting thing about doing this in the scanner with brain stimulation is we can go a step further. And this comes back to the first sort of thing I was saying about causal relationships. And that we can say, okay, yes, there are group mean activity changes, and that's interesting, and they're in the kind of regions we think they probably should be for movement-related things. But actually, instead of just looking at the group mean, we can go on and ask the question, on a subject-by-subject basis, where within the brain does the change in bold signal due to anodal stimulation, which gives us an improvement in behaviour, correlate with that improvement in behaviour? So it's still correlational, but it's on a subject-by-subject basis and saying, is there a direct relationship between brain stimulation and behavioural improvement? Okay. And when we do that, we get a much smaller region of interest, really within the lesioned hemisphere in the primary motor cortex and a bit of the secondary motor areas. And you can see within that really is a very tight relationship. So brain stimulation allows us, combined with MR, allows us to really not only look at group mean effects, but to probe variability in those effects that we all know happens with brain stimulation. We all know that some people respond more and some people respond less. And in this case, we're harnessing that to say, what is that variability telling us? And can we relate that to changes in activity? And because we can do this in the scanner, we can start to ask these kind of questions. So that was an online study. It was an offline study. So we did actually do the stimulation in the scanner, but we recorded before and afterwards. And that's kind of the classic way we've been doing it for years. And that was to allow us to answer a very specific question about where within the brain do we think TDCS is affecting the brain to cause these behavioural improvements. And that allowed us to have a hypothesis that we thought it was primary motor cortex and its lesional side. And we then went on and did, as others have, long-term clinical studies looking at whether or not we can induce a behavioural improvement, having used the combination of brain stimulation and MR to determine where we think we should stimulate. Okay, that's sort of pause point one. Dan, do we have any questions?
All okay so far. So I think we're holding them all up until later on.
Okay, fine. Okay. Excellent. Fine. So that was sort of the first way of looking at it, to look at areas of brain activity and say, can we identify areas of brain activity using this combined approach that we can then target in other ways to induce long-term changes. But BOLD doesn't tell us everything. And it doesn't tell us very much about physiology at all. So the other technique that's very close to my heart is looking at using MR to look at neurochemical concentrations using MR spectroscopy. Okay. So what we do here is use MR spectroscopy, which is the original MR technique. It allows us to look at neurochemicals within a region of tissue. So it's exactly the same as NMR, anyone who did chemistry. We dropped the nuclear bit of nuclear magnetic resonance when it came near people, and everyone got a bit scared about the word nuclear and human in close proximity, but it's exactly the same thing. So what we do is, you can see here, we select a region of tissue somewhere in the brain, For us, we're primarily interested in hand area, primary motor cortex, we're interested in hand function. So almost all the examples I'll show you today are from that region, but this will apply equally well anywhere else in the brain. And we select a sort of 2 centimetre by 2 centimetre by 2 centimetre cube around the area we're interested in. And if we select that region, we activate it and we plot the signal coming back as a function of its frequency, you can see a spectrum here where the individual peaks represent different neurochemicals. So you can see so creatine, NAA, which is a big peak of neuronal mitochondrial activity, and various other peaks. And the height of those peaks broadly gives us an idea of how much of that neurochemical there is in this region of tissue. And this is really just a fit, so LT model fit, which is the commonly used fitting tool for this data, but other fitting tools are available. And what we can do is we can put in the neurochemicals we think should be in the brain, and what it will do is do a linear combination of these basis sets and come up with putative concentrations for each of the chemicals. And in amongst all the things we might be interested in, we can get glutamine, glutamate, and GABA. And I never rescale this slide because I think it is important to say that we can get accurate quantification of GABA, but it's really hard. You can see, I hope you can see my mouse, and you can see that it's really small. It is there, but you sort of have to squint at it. So this is not an easy technique. It does take a long time and relies on people staying very still in ultra high field magnets normally to get it. But we can look at neurochemical concentrations. We can look at various things with them. So one of the big questions we had was if TDCS, or brain stimulation in general, but in this case direct current stimulation, modulates neurochemicals, or sorry, modulates plasticity, does it change neurochemicals? And in particular, does it change GABA and glutamate? So again, this was an old study where we looked at neurochemical concentration in motor cortex before and after 10 minutes of 1 milliamp anodal TDCS to that cortex, compared with a sham control. And again, we acquired the data from this consistent region. And these are the results. And what you can see is that you get a significant reduction in GABA in response to anodal TDCS that's not there with sham control. And this has been replicated a number of times. We don't get a change in GLX, which is our marker of glutamate and glutamine in this case. And that's probably because of the technique rather than saying that glutamate doesn't change. But we do see a significant reduction in GABA. Okay, so this is one of the first studies that used MR spectroscopy to look at the effects of brain stimulation. And that's all kind of well and good, and we were very excited at the time. It was one of my PhD papers, and it was nice to see. We could see changes in neurotransmitters. But it doesn't tell us a huge amount about either brain stimulation or the cortex, to be honest. So the other finding that was floating around was an earlier version of this study. This was a replication of a previous study, which looked at looked at changes in neurochemicals in the brain during learning of a motor task. Okay, and so what we got people to do, we took some young healthy people, popped them in the scanner, and asked them to learn a sequence learning task. So it was a serial reaction time task, a 16 button press sequence that repeated and repeated and repeated. Okay, and each of these blocks you can see on the x-axis were 5 minutes. And as people learnt the sequence, as it repeated enough times, they got quicker at it. And as they did that, the GABA levels went down. So this is the motor sequence group. So motor learning here led to a decrease in GABA concentration. Okay, and that was seen previously as well. And there was no change in either of our two conditions, either when they're just moving their fingers exactly the same number of times or lying there watching David Attenborough. So this then begins to ask an interesting question of, I think, which is, can we use brain stimulation to start to explore changes we're seeing in the brain happening physiologically? Because we've got this decrease in GABA with learning, but can we use brain stimulation to say, is this actually driving the learning? Is this how plasticity occurs in the motor cortex? Or is this just something that's randomly happening? It's an epiphenomenon. It doesn't have any relationship to learning. So what we wanted to do, we did anal TDCS to primary motor cortex, 1 milliamp for 10 minutes, and looked at GABA levels pre and post. We did a control condition to check that this wasn't a global finding. And we did functional imaging during a serial reaction time task on a separate day. Okay. And this design allowed us to ask the question, does a change in GABA, is that causal is a strong word, but is it implicated in motor learning? Does it relate to motor learning? So we get a GABA change when we learn, but actually if we quantify the GABA change on a subject by subject basis, does that predict how much they'll learn? Okay, so can we get a bit closer than just saying these two things happen at the same time to say the amount to which somebody can decrease their GABA on one day in response to TDCS will predict how much they can learn. And it's the GABA responsiveness and the degree to which they can do that that predicts the amount that they can learn. Okay. And it also, which allows us to ask two related questions. One, is it GABA decreases that are important in learning? But also, are TDCS and learning acting on the same circuits? And if they are, at least at some level, and if they are, then that might give us a mechanism for how brain stimulation works. So these are our reaction times. As you imagine, people learn the task, and as they learn the task, the time it takes them to press the correct button in response to the visual cue decreases. We had two blocks in there that were where the visual cues were presented in a random order, and that allowed us to ask the first question we wanted to ask, which was, does your level of inhibition reflect how fast you can respond? Is this a physiological measure of inhibition or not? Does it tell us about behaviour? And it does. So people who had higher levels of inhibition at baseline were also slower to respond, whereas people who had lower levels were quicker. But then the interesting question then is this idea of GABA reactivity, the idea that some people will respond a lot to TDCS, their GABA systems will be really reactive, and are people who are able to modulate their GABA quite easily in response to TDCS, are they also able to modulate their GABA potentially due to learning and learn more? So the ability to decrease your GABA easily, does that correlate with how much you're learning on this task? So we defined learning as just the reaction time decrease from the first sequence block to this plateau, and indeed saw that there is this relationship such that people who have a very responsive GABAergic system, in response to TDCS at least, learn more, as determined by a decrease in reaction times. Okay. So it's not causal. It's not saying that the decrease in GABA is causing the learning, but it is suggesting that there is at least a specific relationship between these two things, and it isn't just the change in reaction times that's driving the decrease in learning for some strange reason. It's that there is actually a relationship between these two things, even when they're happening on different days. So again, combining brain stimulation and imaging and spectroscopy allow us to answer questions about the effects of TDCS, but also to answer basic physiological questions about how the cortex works. Okay. And you can look at whether or not this is specific, and if you look at the learning-related fMRI signal, you see this very specific region in the brain where there is a relationship between learning-related fMRI signal and GABA change. And again, it's primary motor cortex, which is exactly where we'd expect it to be. So this seems to be specific, and it seems to be specific in terms of the anatomy and in terms of the neurochemicals. So again, another way of slightly interesting, hopefully, using multimodal imaging this time and brain stimulation to answer questions about neurophysiology. Okay. But both of those studies had brain stimulation in the scanner, but actually we didn't scan during the direct current stimulation in those cases. But we can do. And that might ask us, rather than relying on the after effects of stimulation, can we use stimulation paradigms that don't necessarily have after effects, but have very definite online effects, and look at what's happening during online stimulation, which is what we'll come on to. How are we doing for questions?
Yeah, could I jump in with a quick question there that I think relevant to that last section? So is the role of the neurotransmitter GABA in motor function known?
No, but there's a lot, and I was sort of skirting around that slightly because I'm conscious that despite my natural tendency to make every talk I give about GABA and motor plasticity, this is not a talk on GABA and motor plasticity, so I didn't go into it in as much detail as I would normally do. But I'm very happy to answer a question like so a decrease, so it is not known, it's still a very much a subject of active study, and it's keeping my group going at the moment, so there's still a lot to learn. The idea from animal models, which seems to be held up by human data, some of which I've presented just now, is that that plasticity in motor cortex is glutamatergic, so it's long-term potentiation like plasticity, it's a glutamatergic phenomenon, but a decrease in GABA is necessary from the animal models to allow that plasticity to occur. Okay. And that seems to be borne up in the human models. There are big questions about long-term learning as opposed to short-term learning, so sort of in stroke, and how that works in terms of other brain regions and how that affects connectivity and other questions. But that would sort of seem to be sort of the reasonably sensible working hypothesis, I guess. Does that answer the question?
Hopefully, yes. We may get a follow-up from that person a little bit later. But I think a related question, I think you kind of just answered it already anyway from a different person in relation to GABA levels and learning specific. Is it specific to the motor cortex, or can we find this relation in other areas as well?
It's a really interesting question. So you definitely can see this relationship in other areas, but it seems to go in different ways, and it depends on the learning. So the primary sensory areas, one of the ways, so if you're looking for signal and noise, for example, in visual system, then actually the way that the cortex learns to deal with that is by putting GABA up to reduce the noise. In some areas, if you're looking for different, if you're trying to extract signal, diffuse signal is difficult to see, then it decreases GABA. And both of those we've demonstrated in collaboration with Zoe Courtesy in Cambridge. So it depends on the type of learning, and it depends on the brain region, but it's certainly not trivially consistent across brain regions.
Excellent, thank you. There is one more question, but I'll keep that until a bit later on because I think it might be relevant to getting into then now the combining of the techniques together. So I'll keep that till later.
Okay, cool. Okay, so yes, so now I've talked to you about a couple of studies that we acquired data before and after brain stimulation and looked at the after effects. I'm going to go on to talk about using brain stimulation actually during acquisition, and a couple of examples of how we might start to use that to answer some sort of fundamental questions about how the brain works. It's important to say that we did do the stimulation in the scanner. You can, of course, do some imaging, take people out, do the stimulation, put them back in again, and that's fine, and it's a lot easier in a lot of ways, but you lose a lot of time, and it's very difficult to reposition people properly. So if you want to look at after effects of these things, you usually want to do that in exactly the same brain region that you looked at your baseline measurements in, and usually pretty closely to the end of the stimulation, and it usually takes 5 or 10 minutes, even if you're really efficient, to put people back in the scanner. So whilst we have done TMS, for example, outside the scanner and then put people in, it actually brings its own issues. So it's not necessarily a simpler way of doing it, though it is possible. But these kind of questions, we were interested in asking actually what happens during stimulation. And one of the things that we were particularly interested in really was, this is a study in collaboration with Peter Brown, who's very interested in beta oscillations. This is oscillatory activity in the motor cortex and in the rest of the motor network, but in this case, particularly in the motor cortex, at about 20 hertz. And this has a big role in pathophysiology. It's very prominent in Parkinson's disease, for example. So in healthy adults, as we go to move, the activity in 20 hertz range substantially decreases, and then it rebounds when we finish moving. And that's classic electrophysiology finding of movement in pretty much any study. And in Parkinson's disease, where people have definite problems with moving, they don't get that characteristic decrease. So we think that it's something important. And there's been a lot of hypotheses about what 20 hertz activity might represent in motor cortex, which again, I'm not going to go into in any detail. But one of the questions was, does 20 hertz activity not only affect sort of the function of motor cortex, but how motor cortex connects with its wider network? So what we did here was use our 3 Tesla scanner. So we're back down to 3T again. And we but a slightly more recent model this time. And what we did was apply 80 seconds of tax with a ramp up and ramp down. Again, not necessarily what I do now, but worked quite well for this. And we did that at 4 periods during the scan. And we just laid people at rest. So this was a resting state scan, and we were acquiring normal fluctuations of bold throughout this scan, and then looking at how those connectivity patterns changed with either 20 hertz stimulation, 5 hertz, which we were using possibly slightly naively, but we were using as an active control to look if there were frequency specific effects rather than just tax effects, and a sham condition as a true control. So the first question we wanted to ask was, if we stimulate motor cortex at 20 hertz, does it change the connectivity pattern of the motor cortices? And it really doesn't. So so this is 20 hertz stimulation, and this is connectivity between one motor cortex and the other, and you can see that there are no changes between sham or between 5 hertz and 20 hertz. So it doesn't change connectivity between the primary motor cortices. We also identified this resting state network, which is identified from an independent component analysis of regions of the brain that are spontaneously, the bold signal is spontaneously fluctuating in a correlated manner. So these resting state networks tell us something about connectivity of a functional network. This one is thought to describe the motor network. So we looked and got a single measure of connectivity within this region for every person in 20 hertz, 5 hertz or sham, and again saw no significant difference, possibly a trend towards a decrease in both stimulation conditions, but nothing significant. My animation's gone all wrong, I apologise. So what we did then was look at the correlation between connectivity of the 2 primary motor cortices. So again, we're stimulating one primary motor cortex, the reference electrode was over the supraorbital ridge, and looking at the connectivity between that motor cortex and the other motor cortex, which doesn't change, and correlating that with the connectivity of the whole of the motor network. And because the motor cortices are prime regions, and there's sort of major network nodes within this network, you might not be surprised to imagine that if you don't perturb the system at all, then there is a very strong relationship between how connected your whole network is and how connected the 2 motor cortices are. And that's what we see, and we've seen that in a lot of studies. And that was unchanged with 5 hertz stimulation. Okay, so we still saw this tight relationship, but actually that relationship completely broke down with 20 hertz stimulation. So it wasn't that it was significantly modulating either M1 connectivity or network connectivity, but that it was disrupting the relationship between those 2 things as we stimulate it. And so, gosh, these really have got messed up, I apologize. And so we built a small model of the motor network, and we've got the primary motor cortices we were stimulating here, we've got the other motor cortex, and we've got the rest of the sensory motor network. And we then used this sort of toy model to ask some questions and said, okay, so what's happening? Can we see differences in connectivity between the motor cortices and how it relates to the rest of the motor network? And when we looked a little bit more carefully, what we saw was that actually, if you exclude motor cortex from the network and you just say, okay, if we take motor cortex out, how is the motor cortices connecting with the rest of the motor network to which they're less connected to start with? And actually you do start seeing a change between primary motor cortex and everything that is not motor cortex, and primary motor cortex and pre-motor cortex. So there is, so 20 hertz stimulation is doing something slightly strange about how the motor cortex is communicating with the other brain regions to which it's connected. So we built this little toy model and to see if it could sort of support what we'd found. If this idea that what we were doing with the tax was taking away these connections, so separating out really how motor cortex was communicating with this motor, with the other motor cortex, taking this primary motor network and sort of separating it from the secondary motor network. And that is exactly what the toy model sort of demonstrated that it probably was doing. So this is the same as the empirical data I showed you, that from this model, that in black in sham and in blue at 5 hertz, there is a tight relationship between the primary motor network connectivity and the whole of the motor network. But at 20 hertz, this breaks down, and it looks like, although there's no overall change in the motor network strength, and there's no change in the primary motor network, the primary and secondary motor networks become separated. And importantly, because a model, especially a toy model like that, can be used to fit any kind of data and give you beautiful results, the model produced a hypothesis that we could then go back and test, which was that whilst there may not be an overall change in connectivity, the degree to which 20 hertz stimulation might change M1-M1 connectivity would be predicted by how strongly the two motor cortices were connected originally. And that would only be true for the 20 hertz and not for the other two conditions. And indeed, we could go and look at that, and we saw exactly that relationship in our fMRI results. So this is really using online stimulation, so not looking after effects, but temporarily perturbing a circuit and asking how does, injecting, in this case, 20 hertz stimulation, what does that do to connectivity? Can we use tax in the scanner to tell us about what 20 hertz activity in one brain region is doing in terms of how that brain region communicates with the rest of the network? And we can go on to look at how it's doing it as well. So this sort of spoke to the idea of the communication through coherence hypothesis, whereby we can see that you get that it can communicate if that brain region A can communicate with brain region C if the spikes are happening at certain times in the cycle. And brain region B is inhibitory, which means that the spikes are coming, the firing pattern from brain region B is coming at this point in the cycle and reducing the activity in C. And in order for these relationships to change, what they need to do, so for B to become excitatory, is it needs to shift slightly so that it's now becoming coming in at the right point and causing activity, and you're getting strengthening of those two regions together. And so we could ask the question, how on earth is that happening, if that is what's happening, and how can the brain change its frequency or its phase to allow this kind of connectivity to change? And so what Magda did, Magda Novak, who was in my group, was take this idea that we've shown you that if we give beta stimulation, we give stimulation at 20 hertz, it will change how motor cortex is communicating with the rest of the network. And she wanted to ask the question, if it's doing that, then is it doing something to neurochemicals? How is it managing to change that activity, and what is changing at the cortical level that is allowing that to happen? And so what she did was record individuals' intrinsic beta activity using EEG, and record the frequency of that outside the scanner. So got them just to do a very simple motor movement and looked at the peak frequency that each individual subject had. And then in the scanner, we did some baseline spectroscopy. We looked at MR spectroscopy during application of either real 20 hertz stimulation or sham, and then we did some post measures. And what we saw was kind of interesting. So when you stimulate with 20 hertz, you get a small but not particularly exciting decrease in GABA compared with sham, and that's mostly driven by an increase in GABA with sham. And we see increases in GABA in the scanner. That's probably nothing exciting, but there's nothing very clear, and it disappeared completely after stimulation anyway. But and there was a significant increase in glutamate, but again, it's really driven by this decrease during, and there's nothing very clear going on. But then she asked the interesting question of, okay, rather than just, again, going back to individual variability, rather than just taking the group mean and going, oh yeah, okay, maybe you do get a change in GABA and a change in glutamate, but there's nothing very clear. Can we look at that in relation to how much the brain activity has got to change to become 20 hertz? If we imagine you've got ongoing activity at, say, 24 hertz, and we apply a 20 hertz stimulation to you, then your brain has to slow down your ongoing activity to match that 20 hertz frequency. And conversely, if it's a bit slower, then it has to speed it up. So if we look at the difference between what your frequency is naturally and 20 hertz and plot that in relation to how much your GABA changes, suddenly we start to see a relationship. So that if you're having to slow down your beta activity, lost my note, then you're increasing your GABA to slow it down. If you're having to speed it up, then you're decreasing your GABA to speed it up. And we don't see that relationship with sham. So again, using online effects to try and understand a very specific question about neurophysiology, in this case, 20 hertz activity in motor cortex. So the first study then looked at what 20 hertz activity does to connectivity to help us understand how that might be changed in patients who have a pathological increase in beta activity. And then to ask a sort of slightly geeky, but hopefully vaguely interesting question about if brain regions change their connectivity by changing the phase and the frequency of the activity in individual brain regions, how are they doing that? What's the mechanism by which they can change that connectivity pattern? And those two questions are really only possible if we start using stimulation in the scanner. Dan, I'm looking at you.
Yes. I have another couple of questions, if that's okay. They are independent questions, but very related to each other. So I'll try and merge them all into one, if that works. So what is the role of the reference metabolite in establishing concentrations of GABA? Would you expect GABA changes depending on the reference metabolite? And the second question related to that was, what's the best compound to normalize GABA with? CR or NAA, and that was kind of like in both.
Yeah, no, those are really good questions. So it's good to choose a reference metabolite that's acquired at the same time, because there's always a risk, particularly with brain stimulation, and we're slightly concerned that we might be causing some kind of shifts in water concentration, for example, that might just shift all our metabolites. So you want to have something that's acquired at the same time so you can see it's a specific change, an increase or a decrease. We used to use NAA, which is not a good reference metabolite. It was the only one we could actually acquire at the time in that particular sequence. Creatine is a good metabolite to use for healthy control studies. It's a marker of sort of cell energetics in general and sort of general cell health. So we don't expect it to change over 20 to 30 minutes of brain stimulation. And if it did, then we'd be quite worried. We'd have never seen it change. It changes with age, and it changes in neuropsychiatric diseases, but it doesn't change in young healthy people over a short period of time. You can calculate absolute concentrations, but you have to have very accurate water measures, and that can be slightly tricky, and I'm very happy to talk to people offline about that. That's probably a little bit of a detailed spectroscopy question for here, but broadly creatine or absolute concentrations are probably the way to go, and it's not clear which is better. Certainly we do them, we look at all our relationships, all our changes in relation to creatine and water, and make sure that it doesn't matter which one you reference it to, you get the same pattern of results. And I think that's probably the sort of neuroscience sensible way of dealing with that question.
Excellent. Thank you very much.
So the final thing I'm going to talk about, to give you some ideas before going on to how you might do this, because I'm conscious of time, is to look at the the specificity of changes. So this is not one of my studies, but it was done at Oxford, and I know it reasonably well. And this is a study by Helen Barron and colleagues that was published in Neuron a few years ago. And what she wanted to do was look at a very interesting cognitive phenomenon and go beyond the bold signal to try and understand what on earth was happening in the cortex, and particularly learn to understand how the cortex learns. So this was a task that relied upon a phenomenon that people have seen since doing the advent of MR, which is if you show somebody a stimulus and then you show them that stimulus again and again and again, you get repetition suppression, and so the brain becomes generally less interested every time it sees the same stimulus. So here, this is stimuli, but if I showed somebody stimulus A again and again and again, your brain might be quite excited the first time it saw it, but it saw a red square. For the fifth time, it's not going to be that excited. And so what they wanted to do was see whether or not you could use that kind of response inhibition, response suppression, to learn something about how the brain learns associations, as this associative memory. So what she did was teach subjects on one day that there are definite associations between these stimuli. And so there were predicted associations between green and red and between yellow and blue. Okay. And so they were trained for a really long time before they went into the scanner that green was followed by red and vice versa, and blue was paired with yellow. Okay. And when they'd absolutely worked out that green and red went together and blue and yellow went together, they then put the people in the scanner. And what they did then was show them a red stimulus and then a pause, and then a green stimulus and then a pause. So you had one stimulus and then you had another one, and that one could either be the one it was paired with, so green and red in this case, or it could be a different one. And the idea behind this was if the brain had sufficiently learnt that red and green went together, then it would treat that as the same stimulus and you get this response suppression. And that was a sort of marker of how the brain learnt it. Whereas if it had the stimulus that it wasn't matched with, say A to D, then it would recognise this as a new stimulus, but it would recognise this as the old stimulus, the same stimulus. And what they were interested in is how the brain learnt that and where the brain learnt that. So they did that study and they looked at activity in the brain in response to the second stimulus, when the second stimulus was one of these and the first stimulus was one of these. So AC, so when you got this one after this one, and it recognised the second stimulus as being quite exciting, you had quite a lot of response to it. Whereby when the second stimulus was the one that it knew was closely linked with this one, you got a much smaller response. Okay, and you can actually quantify this. So you are getting response suppression in response to a different stimulus, as long as it's the one it's paired with, because this brain region has learnt this association. So, but that disappears. Okay, so that happens within a session, and you get this nice response. But if you go back and you do exactly the same thing the next day, you don't get this response. You don't get this pairing. Okay, so even though people still know that A is paired with B, you don't get a reduction in the response. So there must be something that's happening during performance and learning on day one that sort of stabilizes and disappears on day two to remove that response suppression effect, even though the behavior is still there, because they still can get it absolutely right as to which of these ones are paired, because it's not a very difficult task and they spent a long time learning it. So they did a pretty complicated design on 2 days. So they did a lot of learning on day one, so that you really learnt that B was associated with A and that C was associated with D. They then did an fMRI block and then another one, and then people went home. The next day they came back and they did an fMRI block, which showed that they'd still learnt the associations, but that the corresponding suppression wasn't there anymore. And then they did some spectroscopy and some TVCS, and then did another block of fMRI, MRI-tES, and a memory test. And the questions they were really interested in was, if the behaviour that you've learnt, you're behaviourally still just as good at knowing which of these two was associated with each other here as you are here, why do you not get response inhibition here when you do here? And this is really just showing that here you get a a reduction in activity to the paired stimulus here, whereas here you don't. Okay. And they thought that, and the hypothesis they had, which they go into very beautifully in the paper, was that this response suppression on day 1 is due to a specific glutamatergic process. So you get a pairing of the cells involved in these two stimuli, you learn that association, that is a glutamatergic process, you get a change in excitability underpinning that learning. But then overnight, you get a matched pairing of plasticity in the inhibitory system, in GABAergic cells. And so when you come back on day 2, your glutamatergic memory engram is still there, but you can't see it on fMRI because it's matched by a similar inhibitory engram that cancel each other out in terms of overall excitability, meaning that you can't see a change in the fMRI signal anymore, but the behaviour is still there. So what they wanted to do then was see if that was true. So that's a really interesting hypothesis, and it would fit with all the literature, but there's no evidence for it. So what they did then was use direct current stimulation, TDCS, to decrease GABA in this region. As I've shown you previously, anadal TDCS decreases GABA. And what they hoped that would do was decrease activity in that inhibitory engram and thereby release the glutamatergic memory trace that you would suddenly see again in your fMRI, and then you could sort of separate them back out again and reveal that the glutamatergic memory trace was still there, but only if you decreased the inhibition around it. So these are the spectroscopy results. This is the brain region that they were interested in, and they looked at GABA change during stimulation, and what they saw is that GABA decreased during TDCS, as you would imagine. These are the behavioural results. What they see is that in block 1, you get no real difference between the pairs and the unpaired. So you still get a big response to the paired stimuli because you haven't really learnt it yet. By MI2, that disappears. By the next day, there is no difference. Okay, there's no significant difference between the paired and the unpaired stimuli. But if you give TDCS to decrease the GABA, you get that specific repetition suppression back, so that not only behaviourally people can respond to it, but actually it looks very much like day 1. And that can be very closely related to the change in GABA concentration. Okay, so those are the examples. I'm sorry I sort of got distracted by small children and waffled through them. Have we got any more questions about that, or shall I go on to the setup, which I know is also going to be of interest?
Yep, I think we can go straight into the next section, and I've got a few questions to ask at the end.
Fine. Cool. Okay, so hopefully I've taught you a little bit about why you might be interested in doing this. It's a very cool technique. It allows us to ask some questions approaching causality and asks us to ask some very sort of specific questions about neurophysiology that we just couldn't answer otherwise. Okay, and it's really cool, and it's used around the world. But please be aware that just knowing something about MRI and just knowing something about TDCS or electrical stimulation in general does not make it easy to put this into the scanner. Okay, it's potentially really, really dangerous to put electric currents in a big magnet. So please ask an expert, follow the instructions, talk to everybody. There are a few labs around the world, increasing numbers, that have got this set up and are very happy to help. We're certainly very happy to help anybody who wants to get this set up. It is not impossible, but it is tricky and needs a lot of care and attention. There is a paper that we went through some of these details in that might be of use. And then I'm going to talk through a little bit about the major things that you sort of start to worry about in terms of combining these. So there are a lot of things potentially we might worry about, and this is not an exhaustive list, but we worry about current generation in the leads, we worry about heating in the electrodes, worry about sparking, and we've seen that. And we worry about artifacts in the scans that lead you to misleading conclusions. That's not an exhaustive list, as I say. Other things might well happen as well. We have a standard operating procedure that we've worked up over the last 15 years that we are very happy to share with people if they're interested. I don't want to make it publicly available because I don't want anyone to think that it's kind of the answer to everything. It's just the sum total of our knowledge and what we think is important. But we're happy to share it on the proviso that it is not exhaustive, and we would like feedback on things that we haven't thought about that might be an issue. This is the basic design. So you have your electrodes on the head, you have some kind of filter box so that you don't introduce any kind of mains noise or anything else into your data. You then have long connecting cables that goes through the filter tube, so you're not transmitting anything into the Faraday cage, another filter box, and then the stimulator. So this is the sort of standard kit, and the stimulator and the first box stay outside the scanner. These are not MR safe. Do not take them into the scanner. They will attach themselves to it and do bad things. You then have a long bit of cabling with some kind of connection, and then a shorter connector that goes to the electrodes. Okay. Eddy currents are the things that we worry about a lot. So you've got a rapidly changing magnetic field if you're using the gradients, and a rapidly changing magnetic field induces an electric current. And if you've got a loop of wire of any size, especially with the brain in the middle of it, then the worry would be that you would induce an electric current in that loop. And that obviously the worry would be that you're delivering much more stimulation in an unsafe way to the subject's brain. And doing other bad things as well, but that's sort of the primary worry. And so to prevent that, there are a number of things you can do. We're very, very careful not to have little loops, particularly in the transmit receive areas, and you lay the wires straight as much as you can and take them away from the subject and away from the scanner as soon as you can. And the fiber optics in some of the commercially available kits are very useful as well for that. We also, all the kits that are available and everything that's been used has to have very high resistors very close to the electrode. Okay, and that is to prevent any currents that will be picked up in these sort of long bits of copper in the scanner, transmitting them, being able to be passed into the brain. So having some kind of, certainly our kits and Neurocon kits have 10 kilo ohm resistors in them, and that's really to make sure that we don't put current into the brain that we don't know about. We always worry about heating. Again, if you're running electric currents and interact with RF fields and you've got conductive materials in the scanner, there is always a risk of heating. That can be, again, mitigated to some extent, and we have to be very careful about how. To be honest, it's a theoretical risk for us, but we always measure it. So every time we have a new type of electrical stimulation, so if we're doing a new frequency for tax, or we're doing a new current for TDCS, or we've got the electrodes in a slightly different position, or we're doing any kind of different acquisitions or a different EPI sequence or different MR, then we always test for heating. And you can get fiber optic thermodes, and what you do is you pop it underneath the electrode and see what the temperature is. So this is, we mark our electrodes with some kind of oil capsule so you can see where they are, which is always quite useful. And you can see here we just recorded for 10 minutes or so a very, very stable temperature underneath those electrodes. We see a few tenths of a degree of heating underneath our electrodes. We don't see more than that, but we do always check. It is not obvious how the RF and the conducting materials will interact at any given head direction. And so we always test every single configuration before we do it, even though we may have done that sequence or used that electrode placement before. We also use conductive gel, not saline, in the scanner because it doesn't dry out, and it allows us to be surer that we're getting consistent current across the whole of the electrode and not getting hot spots, because that also would be where we would worry about heating. So conductive gel, 1020 paste or something, allows you to make sure that the electrodes continue to work. You don't get drying out, and you don't get patchy connections between the electrodes and the scalp. Sparking is something that we haven't, to be fair, we haven't seen at all with Neurocon kit, but we have seen with other kits. And this has happened when we have got the resistors sometimes, obviously, as with all things, break. And if we put, on one occasion, we put somebody in to the scanner with the electrodes in place, but with a broken resistor. We didn't know about it. We tested them outside the scanner and it worked, but it must have done something in between outside the scanner and inside the scanner. And suddenly we were seeing arcs of electricity. And the participant was completely fine. Obviously, we stopped scanning, got them out. They were absolutely fine, just a bit puzzled as to what this flashy thing was in their eyes. But it leads us now to be very, very careful and check very carefully that we can run current and we're not putting people in the scanner with broken resistors. So the current needs to be able to flow, not only to deliver stimulation, but having a broken resistor that might heat and do strange things very close to the subject is always a bad idea. So now we test the circuit, we test the stimulation outside the scanner, make sure that we can stimulate. We then pop them in the scanner, and before we do any scanning, we just run it very quickly just to make sure it still is running. And then we do our scanning, and then we will do the proper stimulation and things. But what you don't want is a broken circuit in the scanner, because it picks up all kinds of strange things and acts as an antennae and start getting, as you would with any metal in the scanner, you start getting sparks and all kinds of strange things going on. And the final thing then is artifacts to worry about. So we don't have many problems with artifacts. We haven't seen very much, but other people have. And it seems to be dependent on your scanner and dependent on exactly how you route the electrode leads. So we lay them outside the scanner, so we lay them around, we get them out the bore as quickly as you can and keep them away from the subjects. But depending on the interaction between exactly how you're doing that and the type of scanner and the sequences you use, you can see some artifacts. So this is a study in a cadaver done by Andrea Antal and colleagues in Germany. And what they did was just look at signal change during stimulation and saw some bold signal change. This was a cadaver, so obviously this was artifactual. And it does raise some questions as to exactly what we're seeing with the concurrent stimulation imaging. It's an interesting question to deal with and one we're not entirely sure about. As I say, we really don't see it. We've looked at the signal to noise underneath the electrodes. We see slight loss of signal in the bone. So we do get a slight dip in the skull signal immediately underneath the electrodes. But by the time we get back down to the brain, there's no evidence at all for us that there's anything strange going on. But as I say, that does seem to be a scanner and subject dependent thing. And we've certainly worked with other groups around the world to try altering various aspects of their setup and have been successful in reducing the artifacts they're seeing. So they're not impossible to get rid of. They're just a little bit tricky and a bit non-linear. And again, we're very happy, as I'm sure other people are, to try and help with that. But it's something to look at very, very, very carefully before you start actually acquiring data. Okay, so I'm going to finish there. Thank you for your questions. I hope it was useful. I wanted to try and really sort of open up the ideas as to why we can do this and what the interesting questions might be, and show that it might be a way to get a bit closer to causality. And just away from just correlational analyses, which are fine, but don't tell us everything. So electrical stimulation can be combined with MR at 3T and 7T, and that's direct current and alternating current. As long as you test it all, there is no reason to think that this is a problem. And it can allow us to answer questions about neurophysiology, connectivity, and activity very specifically in very elegant studies, particularly in Helen's. It's just lovely. That we couldn't answer otherwise, but it is potentially tricky, and we need to be a little bit careful to ensure that the subjects are safe and that you get the data quality that you want. So I'm going to finish there. Sorry, I went slightly over the hour, but I'm very happy to answer any questions that anyone might have.
Excellent. Thanks very much for that, Charlie. There's the question that I had very early on, which this person has been waiting very patiently, but I think it suits right now, actually. So have you done studies looking at TMS and MRS? Why would you use TDCS versus TMS?
So it depends on the questions that you're asking. First, so there's a scientific rationale, which is they're different things and they do different things. So it depends on which you're most interested in. practically, even though I've gone through kind of some of the issues we face with combining electrical stimulation, it is a lot easier than combining TMS in the scanner, which is a whole different ballgame. And we've never done it. I'm not the person to talk about actually how you do that. But it is, you've got a big lump of metal that you're trying to deliver a magnetic pulse in the middle of a giant magnetic field. It's not an easy thing to do. Whereas electrical stimulation is much easier. So we have used TMS. We have looked at the effects of TMS on various things. We've always done that by scanning, getting them out, doing the TMS, putting them back in again. And that's fine. But there are definitely groups around the world that are doing TMS, but it is really hard.
Excellent. Yeah, I fully agree with that from the technical side of things as well, with like the engineering background to it. But I think we're on the same page there. Doable, but it all depends on how and what and where.
Yeah, you've got to have a question that's interesting enough to be worth the effort. And we never have. So we've never done it.
Then on the topic of 3T and 7T, are there any setup differences between the 3T and 7T kind of TES setups that you've used in Oxford?
Nope. We treat them all exactly the same.
Okay.
And there's no differences alternating and direct current. We test everything exactly the same way. We were very worried about doing alternating current. I was very worried, and speaking as a non-physicist, but our physics colleagues just said, well, it'll be fine. It's no different. If you can do direct current, you can do alternating current. And they were absolutely right. So no, it's no harder at 7T than it is at 3T.
Excellent. So one thing I would add in on that point, the kit that Charlie showed on the screen earlier from the Neurocon setup, which included the filter boxes and the cables, we do know from the technical side of things that that full package is only okay for 1.5 and 3T. What Charlie's group uses in Oxford is among other things, the Neurocon stimulator plus their own solution for the cabling. That's right?
That is correct. Sorry, yes.
But we at Brainbox have had experience then of taking that knowledge and experience from Oxford, and we've helped other labs to replicate the same kind of solution then in 7T as well as 3T. So with the help of Charlie's group, we've been able to kind of help other groups to achieve the same thing. So still, I think the answer you gave there is still true and valid. It's just I would add the bit of caution in there for anybody out there that might have the Neurocon kit in particular. You would need the non-standard version of it to be able to use it directly in 7T. And I think then the loads of people saying thank you, by the way, and great talk again as usual. And there was one question then. It's Rory's question actually, so I'm not sure whether it counts really, whether it's fair, whether he's allowed a question. But no, I think you did answer then in the setup. He just asked about any tips that you would give to dealing with the artifacts produced by the TDCS inside the fMRI. But I think that last section covered all of that, to be honest, and the reference as well that you gave.
Yeah, so it's a really difficult question. So we've never had problems. It's not very easy to predict when you're going to have problems and when you're not. So it's a bit of an art form. It's not. So yes, and I know we've played around with different things, and I've visited other sites, and we've tried, and we've changed various bits of their setup. But like many things to do with RF, many things to do with MR, really, it's not entirely predictable as to what works and what doesn't, and it's really a matter of playing around with it. But yes, as I say, we're very happy to share our experience if that's useful. We certainly have some ideas now of things that are worth trying.
Yeah, excellent. Well, thank you again,